Hi everyone. My topic today is DNA microarray and we'll be talking about the principle, procedure and application related to DNA microarray. And I'll be going through the contents, introduction, principle, technique used for DNA microarray, steps involved in DNA microarray, applications, advantages and disadvantages, conclusion and the reference later. Now, what is microarray? Microarray is a laboratory tool which is used to detect the expression of thousands of genes at the same time and study cells. This helps to determine whether the DNA from a particular individual contains the mutation in the genes. It was developed in the mid 1990s and it was derived from a technique known as Southern blotting. Now, what is DNA microarray? They are actually microscopic slides that are printed in thousands of tiny spots in defined positions, in each spot containing a known DNA sequence or genes. They are known as gene chips or DNA chips. Now, this is how a DNA microarray chip looks. These are the tiny spots that have been printed into them and they have defined positions, and this is where we put the DNA, known DNA sequences or genes. This is known as a microarray DNA chip. Now, let us talk about the history. The history is, it starts long back from when photolithography was found. In 1959, Robert Noyce and Jack Kilby found a process known as photolithography. Now, this technique is a very simple technique in which we use light can to convert it into chemical energy. Now, we use nucleotide arrays onto DNA chips. In 1979, Southern blotting was introduced, and from Southern blotting, dot blotting was developed. Then in 1985, Carey Mullis found out or discovered PCR. Then in 1991, a California-based company known as Affymax produced a first DNA chip. Then Affymetrix, the same company, later dedicated to produce gene chip technology, and it was established in 1993. In 1995 is when they started making DNA chips in the laboratory. Then in 1996, DNA chips that have been manufactured was then used for testing purposes. Then at last in 1999, DNA chips proved valuable for classifying and studying cancers. Later, it was known that it was easy to identify the or distinguish between genes at the same time and many genes at the same time. Now, what is the principle of DNA microarray? The core principle of DNA microarray is hybridization, and this hybridization between the nucleotide. The presence of one genomic or cDNA in thousands or more sequences can be screened in a single hybridization. The property of complementary nucleic acid sequences is to specifically pair with each other by forming H bonds between complementary base pair. The overall method is where the DNA is converted to cDNA and then they are dyed using fluorochromes like CY3 and CY5, and then hybridization is performed. Before getting deep into DNA microarray, we need to know what photolithography is. Photolithography is a process in which we use light to create patterns. This is where here UV light is used. This is the DNA microarray chip. DNA is on it. We use light, and then the other DNA, the complementary bases, attached to them, and a pattern is formed. That is how the photolithography works in DNA microarray. Now we'll go to the requirements. What are the requirements to perform the experiment of DNA microarray? The main material required is DNA chip. Now DNA chip, because that is what in which we perform the experiment. Second is target sample. This target sample is the sample that we put. It can be the cancerous and the normal tissue. Third is enzymes. Enzymes that have been used in polymerizing the reactions. Fourth is fluorescent dyes to dye the cDNAs. And 5 is a laser scanner to know what exactly is happening. Now we'll go directly into the procedure. The first step is procedure is collection of the sample. Now samples, when you collect, we need to collect samples from two different sources. One is, it should be the cancerous one, and the other one should be the healthy cell. So example is where we collect cancerous human skin tissue and healthy human skin tissue. Now we have this sample. After collecting the sample, we need to isolate the mRNA from there. How do we isolate the mRNA? It is extracted either by using a solvent method or a column chromatography. Now solvent method is a simple method where you use phenol chloroform, ethanol, or any other solvents, which in which the sample is put into for a certain amount of days, and we can extract the mRNA. And column chromatography is where we harvest the we lysis the cell, the RNA is bind to the columns, and then we wash the column, and then we evaluate the rRNA. We evaluate the RNA. Now, RNA has all the three RNA. It has mRNA, tRNA, and rRNA. So we need specifically mRNA. Again, we know that mRNA has poly A tail. So we put them into a column that has poly T tails, and this mRNA will bind to the the poly A tail will bind to the poly T tail, and when you run the gel or when you run the column chromatography, what happens is the mRNA is extracted and we can isolate them. Later, we use a buffer in which the beads break down and we get the pure mRNA. Now, the next step is where we need to convert the mRNA into cDNA. The next step is where we convert the mRNA to cDNA. Why do we need to convert mRNA to cDNA? Because mRNA is not stable. They have a problem where they can degrade soon. But cDNA is little more stable. So, conversion of mRNA to cDNA. This is where the enzymes come into play. What happens is we bind them to the the poly T tails bind, and reverse transcriptase enzyme starts the process of forming the template. Now the mRNA plays as the template where the reverse transcriptase copies and puts primers into them, and this copies the DNA strand, and then we get a single stranded cDNA strand. Later, in normal reaction, we continue the process, we get a double stranded cDNA strand, but since we need only single strands over here, we can stop the process over there and we can continue. Then after reverse transcription, what happens is dyes known as cynin 3 and cynin 5 are added. Now till here the steps are common for both healthy and cancerous cells. Fluorochromes are added. Cy, that is green, are added to the healthy cells, whereas C5, which looks red color, it is added to the cancerous cells. Now we have a red color cell and a green color cell. Now the next step to do is hybridization. Hybridization is a process where we put the samples into the gel and we run it into, and we see for, in this process what happens is they bind to the we know that our microarray chips have known DNA sequences. So complementary base pairs in the cDNA bind to the known DNA sequences, and we get the combination of both. Then we have detection using microarray. Now we have a screen, we have a gel gene chip, and this gene chip is kept into a scanner that has a laser. Now the scanner has a laser and a computer and cameras attached to it. So once you keep the slide into them, the light passes. Here photolithography works. The camera records the images produced with the laser and scans the plate. And this scanned image is visible on the computer, and we can store and later analyze the result. The next step is analyzing the result. Now analyzing the result is based on fluorescence. What happens is in the result we can see different color spots. We have yellow, we have red, we have green. Now how is this formed? This is because of the fluorescence, the dyes that we have used. What happens is the healthy cells are seen as green cells, where the cancerous cells are known as the red cells. Now, we have a intermediate color, yellow over here. It says that it has equal rates of being healthy as well as cancerous. So there's chance that that cell can turn into a cancerous cell also, and there's no chance that it can turn into healthy. Now we'll just go through it again. We have two cells. This is the healthy cell. This is the cancerous cell. What do? We isolate the mRNA. After isolating the mRNA, what do? We convert it into cDNA. We got cDNA. Okay, after converting into cDNA, what do we have to do is we have to add fluorescent dyes to them. See, in the healthy cell, we add green cell color dye CY3, and here we add CY red. Then we run into hybridization, and then the results we get this results. Overlaying the result, we get that there's the red and yellow formation. Now, applications of microarray. It is used for gene expression profiling, discovery of drugs, diagnostic and genetic engineering, alternating splicing detection, proteomics, functional genomics, DNA sequencing, and toxicogenomics. Advantages of using DNA microarray is that it can provide data for thousands of genes, and one experiment we have only we can only we should only do only one experiment to get thousands of results, and it is fast and easy to obtain result, and different parts of DNA can be used for studying gene expression. Disadvantage is that the production of it is too expensive to create, and it is lot of time consuming, and the DNA chips doesn't have very long shelf life. Conclusion: the ability to record large number of old samples and analyzing them for various genetic alterations help in understanding the concept of molecular biology. It is used in the analysis for diseases in the oral cavity. And currently, microarrays are primarily a research tool. Microarrays promise a more biologically based, individualized, and vastly improved standard for oral care, where we will have great clinical impact on the way dentistry will be practiced in the future. These are my references. Thank you.